Okay. Welcome to the Youth for Neuro podcast. I'm Anita, a junior in high school and the host of today's episode. Today I am joined with Dr. Shelly Flagel, a research professor at the Michigan Neuroscience Institute of the University of Michigan and a professor in the Department of Psychiatry. Having received her bachelor's degree in psychology, her PhD in neuroscience, and her postdoctoral training all at the University of Michigan while completing numerous projects, Dr. Flagel has gathered immense knowledge and experience in the field of neuroscience. We are glad to have you today.
Thanks so much for having me. Happy to be here.
Mhm. So for our first overall question, but could you explain to our audience essentially what your work as a behavioral neuroscientist includes and what you and your lab seek to study?
Yeah, so my overarching research interest and that of my lab is to hopefully determine the underlying mechanisms in the brain that might contribute to individual differences in vulnerability to mental illness. And so we study that from a perspective of how individuals respond to their surrounding environment and how that maps onto differences in the brain that may then lead to maladaptive behaviors and symptoms characteristic of psychiatric disorders.
Great. And through reading an overview of your lab's research, I found that a major part of your research includes stimulus reward learning and Pavlovian conditioning. So could you elaborate more on that?
Yeah, so I think the simplest way to describe it and to give an example is if you think about using your smartphones and your devices, and you get a notification, whatever that notification may be for you, for signifying that a certain individual is contacting you or that you got a certain like on a post. You hear that tone or you feel that vibration, and you probably immediately have some sort of feeling, and that could be a feeling of excitement or happiness, and that's because you've been conditioned to know that that signal on your phone signifies that what's going to come, and that is, like I said, a message from somebody else. And so that's, I think, the best way to describe it. And all of this is based on what we call classical Pavlovian conditioning studies that were done by Ivan Pavlov many years ago, Russian physiologist. You may have heard of Pavlov's dogs, where similarly they conducted conditioning studies where a sound would signify food reward. And eventually he found, after pairing that sound with the food reward, that the dogs would eventually salivate and get excited just at the sound alone, even when the reward wasn't there.
And how do these concepts relate to your specific research on mental illness?
Yeah, great question. So we found that when we characterize, in my lab, we use an animal model, and it happens to be rats. And when we can put rats into a conditioning chamber where they are exposed to, in this case, it's an illuminated lever, it's a cue. So this lever is presented in a dark box, it's glowing, it's salient, it's attractive. And so it's presented and then it retracts. And right after it retracts, a food pellet is delivered in an adjacent food cup. And so this is, again, classical Pavlovian conditioning, meaning that the food reward will always be delivered after that lever is presented. The rats don't have to interact with the lever at all. They don't have to do anything. Food will always come after the lever is presented. So even though that the food always comes and they don't have to do anything, we see that these very distinct responses emerge. So on one extreme, there are certain rats that will see that cue or that lever, and they go after it, they grasp it, chew on it, gnaw on it, as if it were the reward itself. And so for them, that cue is especially attractive, and it elicits approach behavior, and it becomes what we like to call, you can consider a motivational magnet. I think it's the best way to describe it. And so we call them sign trackers because they're attracted to the sign that signifies the food delivery. And then on the other hand, we have what we call goal trackers, where they see the cue for what it is. It's simply a predictor. Then they go to the food cup when that cue is presented, and they just patiently wait for the food to be delivered. And so by characterizing rats on these individual differences and how they respond to a Pavlovian cue, we found that it actually maps onto a number of traits of relevance to addiction and other psychiatric disorders.
Wow. Okay. And what forms of technology and tools and other models do you use to conduct this type of research?
Yeah, so I'm like you said, I'm a behavioral neuroscientist. So behavior is kind of our major interest in terms of outcome models or outcome measures. But we use a number of approaches to really kind of probe brain function and figure out what brain mechanisms are underlying these differences in behavior. So we use a lot of techniques where we can insert certain receptors into the brain in order to turn on and off certain neurons or brain regions. These are called, the ones that we use are what we call chemogenetic or optogenetic approaches, meaning that chemogenetic, we can insert receptors into the brain and then administer a chemical that will then activate those receptors that then will result in either activation or inhibition of a certain circuit. On the other hand, we have optogenetics, where you can also insert receptors into the brain, but these are reactive to different light intensities. So then we can shine laser light in those certain brain regions and on those neurons and activate or inhibit those neurons or projection areas in the brain. And so those are two of the main techniques that we use to manipulate different parts of the brain and see if that affects behavior.
Wow, that's great. And are there any like new forms of emerging technology that help you with this research?
Yeah, there's a lot. That's one of the really exciting things about neuroscience is how quickly these tools, these new tools and technologies are becoming available, and how they're allowing us to really probe brain function more deeply than we have before. And similarly for behavior. So on the behavioral end, we now can use these certain technologies that allow us to analyze videos using machine learning approaches that allow us to get really fine resolution of the behaviors that we weren't previously able to get with just metrics that were given by the operant chambers. So that's one approach, is using these sort of what I like to call deep phenotyping approaches to really dive into the behavior and see what's going on. And then other than that, there's remarkable opportunities to probe brain function also at a much higher resolution, where we can get information about neuronal activity and put, from a single cell, get that neuronal activity information and put it together with what we're seeing with behavior. And so using these computational neuroscience is changing and becoming more advanced and becoming more important for us to use every day. And so it allows us to integrate, like I said, really fine resolution brain activity with really complex behaviors, and to better understand the underlying neural mechanisms of behavior.
Thank you. And what regions of the brain are most involved in behavior, and what you study specifically?
Yeah, so every brain region essentially is involved in a lot of what we study, but we're focused on what we call the reward or motive circuitry, but also focused on expanding that beyond our sort of classical understanding of what that is. So in my lab, we're really interested in this idea that there are sort of this, what we call top-down control mechanisms coming from cortical areas of the brain. And those, as you may have learned, are involved in sort of executive function and decision-making and planning, and what we call more cognitive mechanisms. And then we have these bottom-up subcortical mechanisms that are more involved in emotional and motivational regulation. And what I'm interested in is how those two things are integrated, and when they become sort of imbalanced. So if you have too much top-down cortical cognitive control, it could become a problem and lead to extreme behavior. Or if you have too much subcortical overdrive, too much emotional motivational overdrive or hyperarousal, that could also become problematic. And so when those things are off balance and not properly integrated, I think it's especially interesting and important to understand in terms of psychiatric illness and symptomatology. And so what we're trying to do, one particular brain region we're interested in is the paraventricular nucleus of the thalamus, which sits really centrally in the brain to integrate this top-down and bottom-up information and then send it to guide motivated behavior. So that's one particular brain region that wasn't really classically on the map in terms of motivation or reward processing, but that we think is really important in this balance of top-down control and subcortical motivational systems.
And for my next question, I read that a key goal of your research is to better understand addiction specifically. So could you elaborate more on this and how your findings connect to the study of addiction, as it is a prevalent disorder in today's society?
Yeah, so we talked a little bit about how the animal model that we use captures differences in how one responds to their surrounding environment, but in particular to cues associated with rewarding outcomes. And so if you think of individuals with addiction, one of the biggest problems in the treatment of addiction is relapse. And individuals with addiction will often report relapsing when they come into contact with people or places or paraphernalia associated with that prior drug taking experience. So when they come into contact with these stimuli or cues, they'll often say, I couldn't help myself, even though I've been abstinent for so long, even though I wanted to remain abstinent, and I recognize the deleterious consequences, I couldn't help myself once put in the face of these stimuli or these cues. And so I think that that's actually what we're tapping into with the model I described, is looking at how individuals respond to cues in their environment associated with reward, that's specifically tapping into this increased propensity for relapse. And so I think that's the link between what we're studying and addiction vulnerability.
Wow. And what underlying processes occur in the brain as an individual develops an addiction, like based on what you've studied?
Yeah, yeah. Also a very complex question, and there's a lot. It's definitely affecting many brain regions, and so this also depends in part on the drug of abuse that you're studying. But in general, if you think about our work and in particular increased relapse propensity, I think it does come to that notion of lacking inhibitory control, so not being able to properly regulate your response, even though you desire, like I said, to remain abstinent, or you recognize the consequences of the potential behavior, but they just can't control themselves. So in that sense, I think what we're studying has a lot to do with these top-down cortical control mechanisms. And like I said, different drugs of abuse can affect certain brain areas or certain neuronal subtypes more intensely than others, depending on that drug of abuse.
Appreciate your response. And our next question is, are there any individual differences in like susceptibility to Pavlovian conditioning? Like what could increase, or I'd say what individuals are more susceptible to this?
Yeah, so yeah, that's a great question. And so I described how we're studying these individual differences in Pavlovian conditioning, and I mentioned that we know that this actually maps onto a number of other traits. So we know that animals that show this increased hyperarousal in response to reward cues that we call sign trackers in the animal model also exhibit a number of symptoms of relevance to psychiatric disorders, which may highlight that there's these other traits that may also signify vulnerability. And so rats that are more hyper-responsive to these cues are also more impulsive. They exhibit behaviors of relevance to attention deficit hyperactivity disorder, relevance to post-traumatic stress disorder, obsessive compulsive disorder, and a few others as well. And so I think my sort of working hypothesis right now is going back to this notion that these deficits in inhibitory control, this inability to really kind of engage your cortex to make the proper decisions in terms of what you're facing at that time, is maybe a common underlying brain characteristic of these individuals.
And what kind of addictions do you focus on most when studying Pavlovian conditioning?
Yeah, so in my lab, we primarily use cocaine, study the effects of cocaine and relapse propensity to cocaine seeking. We've also looked at some opioids as well. And so the way that we usually go about this is that we want to know if these animals that we're characterizing based on Pavlovian conditioning and these individuals do map onto these different aspects of drug taking, but also drug seeking behavior. Like I said, we're really focused on that relapse phase of addiction.
Mm. And describe a typical experiment you may conduct when doing your overall research. So any like experiment.
Yeah, so what we tend to do is we will so we know a lot about this animal model already that I told you about, the sign and goal tracker animal model. And so now what we're trying to do is to figure out the underlying brain mechanism. So we'll use some of those approaches that I told you, where we'll insert receptors into the brain to be able to really, kind of hone in on a particular brain region or brain pathway. So for example, we might look at that top-down control mechanism by specifically targeting neurons projecting from the cortex to the thalamic region, that PVT region that I told you about. And so we can target that area where we can insert receptors in there that are then going to turn on this top-down control pathway. And we can ask, well, if we turn on this top-down cognitive, these cognitive processes, are we able to inhibit those tendencies? And so we would look to see if we insert the receptors, turn on this pathway, does it affect that tendency for the Pavlovian conditioning, that tendency to sign track or to attribute this excessive incentive motivational value to the cue? And then if it does, then we might ask, okay, well, if we manipulate the same brain pathway or circuit, does it affect other aspects that we know are related to sign tracking? So does it affect impulsivity? Does it affect relapse propensity to cocaine seeking or opioid seeking? And that's typically the way we do it in our lab, and we move kind of around the brain. We have a blueprint of our hypotheses of what brain regions and circuits are involved, and then we move around the brain to see if we can identify whether or not they actually are.
Thank you. And what methods have you developed to potentially counter addiction in the models you use?
Yeah, so we're not using simple pharmacological approaches that will necessarily be immediately transferable or translatable to human use in clinical studies. But I think by understanding the neural mechanisms and the circuitry that we're actually not too far away from being able to manipulate the same circuits in humans. So I think we're closer than we ever have been before, and there's a lot of different methods that we can use in humans now to, for example, stimulate certain brain regions. And so these are used more commonly in neurodegenerative disorders like Parkinson's, but also are starting to be used in psychiatric disorders as well. And so if we're able to identify certain brain regions or pathways that we think are important to be activated or inhibited in order to better control behavior, then I think this is a way that we're going to help to translate treatment options for human use.
And my last question is a little more general, but what's the most exciting part of your work and what motivates you to continue it?
Oh, great question. Yeah, I mean, I'm constantly learning, and that's what I love about, with all of these new technologies and approaches, just in terms of staying up to date on that, that's a lot to learn and stay on top of, and I think it's also so exciting. And so I think figuring out how to best use those tools and technologies to truly advance our scientific knowledge rather than just use the fancy techniques to do what has already been done. That's really exciting to me is to really take advantage of these new approaches in a really sort of cognizant and way and thoughtful way is really exciting. I think what I love most about my job also is kind of the mentorship and being able to work with students at all and trainees at all levels as well. So undergraduate, graduate, and postdoctoral trainees, and knowing that they're going to have a huge impact on our field and on the future of our field is a great feeling as well.
And that leads me into my final question, but what are your future goals in terms of your work and your research?
Yeah, I mean, I kind of told you the overarching theme, which is I truly do hope that ultimately we'll be able to really advance our knowledge pertaining to the brain mechanisms underlying psychiatric illness and hopefully be able to use that knowledge to have it translate into better therapeutic options for those suffering from psychiatric illness.
Mhm. Well, thank you for joining us today, Dr. Flagel. For our listeners, be sure to look into her work and publications if you are interested in learning more. And if you enjoyed this podcast episode, be sure to give us a follow at Youth for Neuro on Instagram, Spotify, LinkedIn, and check out our website linked in our bio. Until next time, this is Anita signing off. Thank you for being with us.
Yeah, thanks so much for having me.
Mm.